Okay, so I guess we'll go ahead and get started. Thanks everyone for coming to our AMS Colloquium. So today we're very pleased to have Dr. Arthur Sherman here from NIH to give us a talk about some of his recent modeling work in diabetes pathogenesis.
Okay. Well, thank you, Cecilia, for the invitation. I've had a lot of fun. I made a lot of new friends, including at the medical school. Happy to see some of them here. And so what I'm going to be talking about today is partly physiology and medicine and partly mathematics. Hopefully there's something for everybody. Specifically on the mathematical end, I'll be talking about diabetes pathogenesis as a threshold crossing process. And I want to acknowledge my postdoc, Jun Ha, who did all this modeling work, and this absolutely would never have happened without him. Okay, so what are the questions that we're going to consider? Type 2 diabetes, T2D, is a disease of unregulated glucose, or hyperglycemia. So the main question is, why do people get diabetes? Well, we know that most people with diabetes type 2 are obese. However, most obese people never get diabetes, only about 20 or 25%. So what distinguishes these two groups of people? All right, so as background, let me just briefly introduce the main organs that regulate glucose in the body. So one of them, surprisingly, is the brain as a consumer of glucose. Even though it's relatively small, it consumes 20% of the glucose in your body. And one of the other organs, the liver, is a special store. It does many things, but in terms of glucose, it stores glucose for in-between times when you're not eating in the form of starch or glycogen. And then when you're in fasting conditions, it breaks that glycogen back down to glucose for the special use of the brain. During that time, other energy-consuming organs like muscle do not use glucose because there's not enough insulin around for them to take glucose up. Instead, they rely on fat, which is stored in the fat cells. Finally, when you eat and glucose enters the gut and then the bloodstream, it goes to the pancreas, to little mini organs called the islets of Langerhans, which are filled with insulin-secreting cells called beta cells. And they have other endocrine cells, alpha cells, delta cells, but no gamma cells, that secrete other hormones. But we're not going to have time to talk about those today. Nonetheless, we have done some modeling on them, and they're very interesting. Well, when they see glucose, they secrete insulin, and then the flows of energy and fuels change dramatically. With insulin present, muscle is now able to take up glucose. The brain doesn't need insulin, so it continues to use glucose, but there's enough to go around for both purposes. The liver, instead of releasing glucose, now stores it. Specifically, insulin will suppress the release of glucose and promote the uptake of glucose. And then fat cells, instead of releasing fat for use as a fuel, will turn around and become storehouses, and they will take up the fat instead of releasing it until the next time that you're in fasting conditions. Okay, so from an engineering or mathematical point of view, this is a classic negative feedback loop, or in biological terms, a homeostatic feedback loop. Glucose stimulates the insulin secretion by the beta cells, and insulin promotes uptake of glucose by the tissues. So that brings the glucose back down, and then the beta cells turn off, and you're back to where you started. Okay, so how does the system break down and result in type 2 diabetes? The standard model is that people become overweight or obese, and that makes them become insulin resistant, meaning, yes?
What about the Japanese who aren't insulin resistant?
Okay, so that's another more complicated story, but they have more beta cell defects. so that they are much more prone to diabetes even at very low levels of insulin resistance. But insulin resistance probably still plays some role. If the beta cells are really bad, then you can become diabetic without any insulin resistance. And one class of diseases that looks like that is called MODY, which is maturity onset diabetes of the young. They develop a relatively mild diabetes just because the beta cells don't work. But those are rare cases. Most people go through this path. Even, I would say, even in East Asia. We could talk about that.
Yeah, we should talk about it.
Okay, so obesity leads to insulin resistance. And then the question is, do the beta cells compensate by secreting more insulin? If they do, then you end up with high insulin, but normal glucose, hyperinsulinemia. And it's not a very healthy situation, but at least you're not diabetic. Obese, hyperinsulinemic, non-diabetic people have a twofold increased risk of cardiovascular disease, but diabetics have a fourfold increased risk. So that's worse. And what happens if the beta cells do not compensate? Then the glucose becomes high, and that's the definition of diabetes. But I didn't say anything about what happens to insulin in that case. So let's look at a little bit of data. So this is a case study from a strain of rats which was bred to be prone to obesity and diabetes, the Zucker diabetic fatty rat. So this is what the glucose looks like over time, and I'm only showing the females because the males are a little bit more complicated. If you put them on a normal diet, low-fat diet, then their glucose basically stays in the normal range. But if you put them on a high-fat diet, then the glucose starts to rise, and then after a few weeks, it starts to rise very rapidly. So there's kind of an inflection point here. And if you follow the insulin during the same time period, then you see that the insulin in the rats that are destined to become diabetic actually goes even higher than it does in the low-fat rats that stay normal. Nonetheless, this increased insulin is not sufficient to control the glucose, and then eventually their ability to secrete insulin is compromised, and insulin actually goes down. At the end of the 6-week study, they have about the same insulin as the low-fat rats, but their glucose is much higher. So this pattern is sometimes called Starling's law of the pancreas, by analogy to completely unrelated physiology that happens in the heart, where something goes up and then comes down. But this has confused people quite a bit, because if diabetes is a disease of inadequate insulin secretion, then here we see the pre-diabetic and then diabetic rats actually have higher insulin than the normal rats. So that has led to the development of an alternative model. It's actually an old model. You can find it in the literature in the 70s and 80s, but it's had a comeback recently. And I believe it's partly because of people's frustration that we're not doing a better job of solving the problem of diabetes. And so maybe it's because our ideas about diabetes are just wrong. So they propose the following, that you have a primary defect of insulin hypersecretion for one reason or another. And that leads to, on the one hand, insulin resistance. So the body has negative feedback that makes it resist the rise in insulin. You become insulin resistant. And there's also the idea that high insulin helps you become overweight. Well, as I mentioned, insulin helps you store fat. So obviously, if you have more insulin, you can store more fat. And all the low carbohydrate diets that have become popular over the last 10 or 20 years are based on this principle that high insulin drives weight gain, so therefore you should try to keep your insulin down by consuming as little carbohydrates as possible. So the hypersecretion gives you insulin resistance, which is the opposite of what I was saying in the standard model, where the insulin resistance induces compensatory high secretion. At the same time, the hypersecretion causes the beta cells to work too hard, or something like that, and then they fail. I'm hedging a little bit because these theories have not been written down quite in a rigorous form or in the form of models, so it's hard to know exactly what the target is, but it's some combination of these ideas, and the combination of insulin resistance and beta cell failure leads to diabetes. Okay, so let's do some modeling to try to see if we can say something about this question. So the most basic representation of the glucose-insulin negative feedback loop would go like this. We have a glucose equation, an insulin equation. The rate of change of glucose depends on hepatic glucose production, glucose that comes in through the meals, and uptake by other tissues, mostly muscle. We also have an insulin equation, which is the balance between secretion and clearance. So before we even break these terms down into their components, we could already say a few things. If you want to maintain normal glucose in the face of a decrease in uptake, that would be insulin resistance, or increase in hepatic glucose production, which would be insulin resistance at the liver, as opposed to the muscle, which is mainly uptake, then either secretion must increase or clearance must decrease in order to bring you back into balance. And probably both things happen, but we're just going to focus on secretion today. So that's the compensatory high insulin secretion idea. On the other hand, if secretion increases for some reason, well, if you do nothing else, then that will drive your glucose down and you'll actually become hypoglycemic, which is even more dangerous than being hyperglycemic. It can kill you in minutes or hours rather than months and years. So a way to fight the high secretion, if that's the primary event, is that hepatic glucose production must increase or uptake must decrease. In other words, you become insulin resistant to defend against the high insulin. Okay, so now let's get a little bit more specific. And these equations are adapted from what's called the minimal model, which was developed by Richard Bergman and Claudio Cobelli, and is in wide use clinically to evaluate the metabolic status of patients. There are people in this room who use it. So in particular, the uptake term is broken up into two parts. One is insulin dependent and one is insulin independent. The insulin dependent term is modeled as being linear, even though it's really not linear, but linear is good enough for our purposes. And the coefficient in front of it, SI, means sensitivity to insulin. So this product, S times I times G, is going to give you the rate of uptake of glucose into muscle. And then there's this background rate that doesn't depend on insulin. And for the insulin equation, we have the secretion term and the clearance term. Clearance is just a simple first order removal. And the secretion is broken up into several parts. So we have, I already told you about SI and I. Now we have a sort of basal rate, or not, a fundamental rate of secretion of insulin depending on glucose, and that depends on how much insulin is secreted per cell. We call that sigma, and how many cells you have, or you could think of it in terms of mass, but it's the same thing. And then we have the volume of distribution that the glucose goes into. So if we want to do both humans and rats or mice, we have to change this volume of distribution. So already this trivial equation is telling you, for example, that if V gets bigger, then beta has to get bigger. And in fact, the masses of beta cells in different species roughly scales with their volume. Okay, so if you have insulin resistance, that means SI goes down, but I increases proportionally to compensate, then SI will remain constant, and then glucose will remain the same. On the other hand, if SI as a product goes down, then glucose rises, and that's the condition that leads to type 2 diabetes. So that is a mathematical way of saying that the compensation is inadequate. And this product, SI times I, although often expressed in different but similar forms, is called the disposition index. Although oddly, if the disposition index goes down, that means you're more disposed to get diabetes. Could be confusing. Okay, so then the question becomes, how can you raise insulin if SI goes down? Well, you can do it by raising mass, that's beta, or increasing function, sigma, or reducing clearance. All those things happen. Okay, so the next step beyond this was to go beyond looking at a snapshot of what a person looks like at some point in time and say, how could this process evolve dynamically over periods of months and years? So this was a model that was developed by Brian Topp and mathematical colleagues, physiological colleagues at the University of British Columbia in the year 2000. And they added a third equation. So the glucose and insulin equation, which I already talked about, describes events happening on the fast time scale of meals, that is hours, whereas the slow time scale is the time scale over which diabetes develops, which is weeks in rodents, but months and years in humans. So that equation looks a little complicated, but it's really just an exponential growth law. So we have the rate of change of beta cell mass is proportional to beta with a time constant tau beta, and there's a function multiplying it which depends on glucose, which I've broken up as just proliferation and apoptosis as a function of glucose. So the balance of these two determines whether beta goes up or goes down. And these things could be very complicated, but that's the basic idea. Now, part of what they propose, and the key part that made this work, is that when you have modest rises in glucose, then the main thing that happens is that P goes up, or P goes up more than A goes up, so that you have an increase in beta cell mass. But if glucose continues to rise, eventually you get to a point where A increases more than P, and then the sum is negative, and then beta starts to go down. So initially you have negative feedback where the rise in glucose is combated by increased beta cell mass, but if you get too high, then you get positive feedback, a runaway system, and then in the simplest case, mass will actually end up going to zero. So there's nothing about beta cell function in here, which was in the model as a parameter, but it was not a dynamic variable. Everything was based on mass. So this model was kind of neglected for a number of years, but recently it has started to get a lot of attention, and there are many groups that have come up with different versions, all based on this same principle of negative feedback tipping over into positive feedback. So we have models by Andrea De Gaetano in Rome, Pranay Goyal in Pune, India, and several others that are similar. Okay, so let's go back to our case study of the ZDF rats. And the first thing that June and I did when we got into this business was to see whether the top model, which seemed very reasonable to us, could be made to fit this data, which actually was also collected by Brian Topp. He did the experiments as well as the modeling. So I already showed you the glucose, but there's at the same time as measuring glucose and insulin, they also measured the insulin sensitivity in an approximate way for the low-fat and the high-fat rats. And what they found was that the insulin sensitivity dropped more rapidly and to a lower level in the high-fat rats. So both groups of rats were insulin resistant, but the high-fat rats were more insulin resistant, and that's the difference. So our hypothesis is that that's the only thing that you need to explain the different behavior. There could be other effects of consuming excess fat, but we didn't need to use those. Okay, so here's the glucose trajectory that you want to explain. The slow rise followed by a rapid rise in the high-fat rats. The insulin, which peaks and then comes down, but is over this experiment higher in the high-fat rats. They also measured beta cell mass. Unfortunately, it meant they had to kill the rats. We really need a non-invasive way of doing it, but for now, this is some of the best data we have. And what they found was that mass actually went up a bit in both groups, and it went up a little bit more in the high-fat rats, but again, it was not enough to prevent the diabetes. And finally, they also estimated the function. For the low-fat rats, it stayed pretty much constant. For the high-fat rats, it went up, and then it came down. And it looks like it went up first, and it came down first before mass. So it seems like function is playing more of a role of actually driving the animals to diabetes. Okay, so those are the things we want to explain. So we found that we had to build in more machinery and take into more features of the beta cell physiology to explain this. So there are actually multiple mechanisms by which beta cells can respond to glucose. The first thing that happens is that glucose comes in, it goes through glycolysis, goes into the mitochondria, and leads to the production of ATP. The ATP-ADP ratio goes up, and that closes a special class of potassium channels called the KTP channels. When they open in low glucose, they keep the cells electrically silent. When they close, the cells can depolarize, membrane potential goes up. That allows voltage-dependent calcium channels to open. Calcium comes in. The calcium then triggers the release of vesicles. The third step is that if you keep the glucose up for a while, like the duration of the meal, that some of the vesicles that are released have to be replaced. So the third step is more vesicles going to the plasma membrane, and then they undergo some biochemical transformations, and then they get into the readily releasable pool, and then they can go out. So we have 1, 2, 3, 4, and the fourth step is when all else fails, you make more beta cells. If the individual cells can't increase their secretion enough, you start replicating. So we put all these features into our model. The glucose metabolism and closure of KTP channels is what happens every time you eat, and you go up this dose-response curve. So this shows the secretion per cell versus the metabolic rate in the beta cells, but you could just think of this as glucose. So you just go up and down this curve with every meal, and if you never become insulin resistant, then it'll be that way for your whole life, and we don't need to consider anything else. But suppose you go to a conference or you go on vacation and you start overeating, well then you become transiently insulin resistant, and the easiest thing that the beta cells can do is reduce the expression of KTP channels in the plasma membrane, which shifts this dose-response curve to the left. So then you get more rise in calcium for the same rate of metabolism and for the same amount of glucose. And that increases the secretion. But suppose, and then when you come back from your vacation, you go back to your normal eating, and your insulin resistance will go away, and this curve will shift back to the right, back to where you were. But suppose you gain weight steadily over a period of months and years, and the insulin resistance does not go away. So we propose that the next step is that you can increase the total capacity of the beta cells to secrete independent of calcium. And this actually corresponds to a known mechanism that we have studied separately in beta cells. When you metabolize glucose, there's some factor that has not yet been definitively identified, released by the mitochondria, that increases the efficacy of calcium. So for the same calcium, you get more secretion. So mathematically, that ends up looking like increasing the maximum of this curve. So this is like the Km of an enzymatic reaction, and this is like the Vmax. And finally, the last step is to replicate your beta cells. So steps 1, 2, 3, and 4 are all included here, and we think of them as happening sequentially in time. Fastest, a little slower, slower, and slowest. Okay, so when we put all these pieces together, we end up with a system of 5 equations. So we have glucose and insulin from the fast system going all the way back to Bergman and Corbelli. We have the mass equation for beta that we inherited from the top model, but we also added this faster step, which we call gamma, control of the shifting the dose response curve, KDP channels, and finally another slow variable, but not quite as slow as beta, is function. And we made a similar kind of equation that has both positive and negative feedback in it. We don't really have much data to support that, but it gives us the result we want. So there's probably whole careers of people to go digging into each one of these mechanisms to explain how they work or find better ones. But for now, we're just going to go with this. Okay, so does the model work? Well, not surprisingly, it does. Here's the examples. Here's the glucose, the high-fat rats, the low-fat rats. We see the slow rise followed by the fast rise. Here's the insulin. It goes up and comes down. It's higher in the high-fat rats. Here is the beta cell mass. It goes up and comes down. Here's the beta cell function. It goes up and comes down with bigger swings and more rapidly. And finally, this is the beta cell function in terms of shifting the dose response curve. So when this goes up, then the dose response curve shifts to the left. We get more calcium entry. So this was a prediction of the model. And then we did experiments with our colleagues at the University of Michigan, taking islets out of a mouse and exposing them to high and low levels of glucose in vitro overnight. And what we found, and our assay was to look at calcium oscillations. I had a whole career modeling calcium oscillations before I did this stuff, and they had a whole career measuring it. So this was a good assay for us to try different steps of glucose, and the end result is that the duty cycle of the oscillations increases in the islets that are exposed to high glucose. So if you plot the duty cycle or plateau fraction versus glucose for the islets exposed to low or high glucose, the high glucose shifts it to the left. So that is roughly compatible with what we predicted. Okay, so let's go back to the original question that I posed, which is the standard model versus the alternate model. So, to repeat, the standard model says that insulin resistance leads to beta cell failure. The alternate model says that hypersecretion leads to insulin resistance and beta cell failure. So let's see how the model stacks up. Now, one of the arguments that's given for the alternate model is that hyperinsulinemia appears long before glucose rises. But our model shows that the standard model actually can reproduce this effect. So that doesn't mean the standard model is right, but is not falsified by this observation. A second argument for the alternate model is that insulin resistance does not change much immediately preceding diabetes. It tends to go down, saturate, and then diabetes happens later. So the argument is, how can that be the proximal cause of the conversion to diabetes? But again, the model shows, as well as the data, that the insulin resistance goes down and saturates, and then, not showing on this picture unfortunately, then the glucose rises. So this is also compatible with the standard model. Now, both of these features are explained by one simple mathematical idea, which is a threshold based on bistability. So a cartoon of that would go like this. You have a curve that says what your steady state glucose would be as a function of how good your beta cells are. So as you go to the right on this axis, then you have worse beta cells, more beta cell defect. And you start off life over here in a condition of health, that's low glucose, but when you develop insulin resistance, glucose tends to rise. Now, if glucose doesn't rise too far, you stay below this curve, which is a threshold, then the flow of this dynamical system is taking you back down. That is, the negative feedback of beta cell compensation will tend to push your glucose back towards a normal state. So transient obesity will relatively easily be resolved by increased insulin secretion. On the other hand, if you cross the threshold, then you have positive feedback. So then your secretion gets worse, you lose beta cell mass or your beta cell function declines, and that causes your glucose to get even higher, and then you end up in the disease state. Now, the threshold here is drawn suggestively around the diagnostic level for diabetes, which is 125 milligrams per deciliter. That number was arrived at empirically by looking at what level of glucose people started to develop complications like retinal dysfunction or kidney failure. But what the model says is that this is actually a dynamically defined point where negative feedback changes to positive feedback. It also suggests that the threshold is probably not horizontal. That is, the threshold is probably not the same for all people. And some people live over here where they're far from threshold and they are less at risk if they gain weight. Some people live over here close to the threshold and they're at higher risk.
Isn't the threshold for where they call diabetes now and what does damage, that's changed, right? Because nowadays, the artificial pancreas people have started saying, if you have an A1C that's below 7.5, you don't see any negative consequences anymore, which is more like 150.
Well, maybe that's because they can't do better than 150. I don't know. But we tuned our model to have a threshold around 125.
Got it.
If somebody tells us it's 150, we can accommodate that. It doesn't change the main story.
Yeah, 125 is appropriate for type 2 diabetes. The artificial pancreas is patients with type 1 diabetes, and you have to worry about hypoglycemia. So they're finding a balance, but 125 is for type 2.
Okay.
I mean, that is what they say, but when you look at consequences for people with type 1 or type 2, the consequences show up after 7.5. For people with type 2, too, also.
Well, 6.5 A1C predicts complications in type 2.
Okay, so
Yes.
let's argue about
I would
that.
vote 125.
Okay, so what was the point I was trying to make about this? All right, so different people are at different distances from threshold, different levels of risk, and yesterday somebody was asking me about gestational diabetes. Gestational diabetes on this diagram more or less corresponds to this curve here shifting to the left, which basically moves your threshold down. And if you get diabetes during pregnancy, that is unmasking the fact that you're probably living close to threshold. So it means that even if the diabetes resolves after the pregnancy, that you now know that you have to be very careful for the rest of your life. So anyway, this very simple picture already tells you a lot of stuff. The actual bifurcation diagram of the model looks like this. And a lot of the work that we did modifying the top model was to change this shape so that this was closer and that the flow was very slow over here, because the top model, people tended to develop diabetes too rapidly. So they had the right idea, but it was quantitatively a little off. So by putting in more variables and more parameters, we could refine that. But the ideas are essentially the same. Okay, so to summarize, the threshold concept resolves most of the issues that I raised in the beginning. It explains the delayed but sudden rise of glucose after a slow rise of insulin. It explains why prevention is easier than cure. So in our first paper, we talked about that this is a possible explanation for why bariatric surgery is, why diet and exercise is ineffective after you become diabetic, even though it's quite effective before you become diabetic. Now, my institute invested millions of dollars to study this in the diabetes prevention program, and they found that a modest diet and exercise program, people losing about 10% of their body weight, could dramatically reduce the rate of conversion to diabetes by 2/3 in an at-risk population. So they said, great, let's do this with people who already have diabetes. And it was a miserable failure. Only a small fraction achieved remission, and most of them relapsed after the first year, also because they gained the weight back. Well, the model says it's because once you've gotten diabetes, you've crossed a threshold, and diet and exercise is not enough. But extreme interventions like bariatric surgery, we argue, are capable of pushing you below threshold, and then the body's natural healing properties or natural healing negative feedback can take over. Okay, so I want to spend the remaining time doing a little bit of math and look more closely at this issue of threshold, because actually I led you up the garden path. The explanations I gave are correct as far as they go, but when you look more carefully, you see that they don't hold up. So let's look at a case where it does work. So let's consider a slow versus a fast drop in insulin sensitivity. So remember that the high-fat rats in our rat study became insulin resistant more rapidly. So if you have slow insulin sensitivity drop, so that's the black curve, then there's enough time for insulin secretion to increase because beta cell mass is increasing. And actually what we're using here is the top model for mathematical simplicity. But the principles should apply to the full model as well. If you have a rapid drop of insulin sensitivity, then insulin first increases and then it comes back down, and beta cell mass does the same. And we see the glucose goes up, and then finally it goes very high. So if you look at this on the bifurcation diagram, it works out. So if you have slow drop in insulin sensitivity, then the glucose goes up, doesn't hit the threshold, and then eventually mass increases and the glucose comes back down. Whereas in the case where the insulin sensitivity drops rapidly, then you cross the threshold and the glucose goes all the way up to 600. So chalk one up for the bifurcation diagram. But let's consider another case, a transient rise in glucose. And this actually happens every time you eat. That well, this is extreme. These numbers are not realistic. But in our more refined model, you actually can get your glucoses above the threshold level just by eating a meal. So here what we see happens is that the glucose goes up above the threshold, but then it comes back down. So here's the glucose in time, going up and coming down. Let's see, what's different about this case? Oh, this is transient insulin resistance. A person becomes insulin resistant for a short time, glucose goes up, but then the insulin resistance resolves and the glucose comes back down. So again, we see it go above threshold, but it's harmless, and you come back down. Well, the key thing is that these rises in glucose were transient, so you didn't stay above the threshold for a very long time. So if you want to understand this, you have to look again more carefully at the equations. So here's the top model again, and what you can see by looking at the beta equation is that the steady state glucose is independent of SI. SI only appears in the glucose equation. It does not appear in the beta equation. So that explains why, that says why the bifurcation diagram does not explain the transition to diabetes, because the glucose steady state is determined by the beta equation, and then once you know that, then you can go into the glucose equation and get I, and finally you can go into the I equation and get beta. So everything is determined once you know the status of the slowest equation. Okay, so in order to understand this, we have to plot the slow manifold, and the simplest way to do this is to look at the G-beta plane. So what we've actually done is added, you have to add as well. Let me come back to that point later. Let me just show you the diagrams. So here is the slow manifold, showing beta versus G, and along this manifold there are three steady states. The leftmost one is the healthy state, the rightmost one is the diabetic state, and the in-between case is the threshold. So there's a one-dimensional flow along this manifold with the arrows pointing towards the stable steady state, and then on the other side of the saddle it goes towards diabetes. So when you have a transient rise in glucose, the glucose jumps to the right, but then the fast flow takes you back to the slow manifold, and since you haven't had time for beta to change, you still haven't moved off the stable steady state, the healthy state. You just, or maybe you've moved a tiny bit, but you haven't gone past this threshold on the manifold. So you're okay. Now let's look at the small step in SI. Well, a change in that, this is basically the glucose equation. And if you reduce SI, that shifts this curve to the right. So now glucose will jump over to the new position of the slow manifold, but you're above the threshold, the X. So you move back up to the stable, healthy state, and the model predicts, or this picture predicts, that you will have higher insulin and high beta, but your glucose will end up being the same. Okay, but suppose you have a big step in SI, you become very insulin resistant. Now this manifold moves further to the right, and so you end up below the X, and that means that you're caught up in this flow. I didn't redraw the arrows, but the flow is pointing towards the diabetic state, so then you descend down the curve to diabetes. So it explains the difference between a little bit of insulin resistance and a lot of insulin resistance. And finally, if you have a big step in SI but it's transient, well again you have the manifold shifting far to the right, but you don't have time to travel down to the diabetic state, and so when SI goes back up to the normal case, this curve shifts back to the left and you jump back before you have time to get caught into the basin of attraction of this point. Okay, so the importance of this physiologically, as I mentioned before, is that it's very easy to go above the nominal threshold. You can go above 125 and not be anywhere near diabetic, as long as it's transient. So that means that the slowness of beta is a double-edged sword. On the one hand, it's protective against these transient excursions above threshold, but on the other hand, if you have someone who's diabetic and you're trying to reverse their diabetes by increasing their beta cell mass, well, beta cell mass changes very slowly, if at all, in adults. So it makes it very hard to combat diabetes.
When you say the 125 threshold, I just want to make sure I understand. Is that, are you talking about the mean over time, or are you talking about the peak value?
Transient. The transient during the day.
So the peak.
The peak.
Okay.
So the naive interpretation of the bifurcation diagram is that the postprandial peak is already putting you above threshold, so why don't you go up?
Yeah, because it happens a lot.
Right. But the point is you don't stay there for very long. You can violate the law as long as you don't get caught. So another way of thinking about this mathematically, so if you have been exposed to other problems involving thresholds, like nerve action potentials, which involve crossing thresholds, or equations like the Ficci-Nagumo equation, which look very much similar to this and are no more complicated than this, that threshold is in the fast variable. This is a threshold in the slow variable, so that's why it has this behavior, and you have to look at the slow manifold. Okay, well, the proper way to do this is to actually look at it in 3 dimensions, and to augment the system by adding SI as an additional slow variable. So then you have two fast variables, G and I, and two slow variables, beta and SI, and then you can make this slow manifold surface and project the trajectory. So I was showing you I was showing you the G beta trajectory, but beta is actually proportional to I in the slow steady state, so really looking at this plane. Anyway, you can go through all the details in 3D, and it's basically the same, and different planes are better for explaining different phenomena. Okay, so let me summarize. We have shown that much of the data used to support the alternate model is consistent with the standard model, and I repeat, that doesn't mean the standard model is right, but it means the arguments that have been raised so far, and the data we have so far, do not invalidate it. In particular, insulin rises before glucose. Insulin resistance saturates before type 2 diabetes appears. And the simple idea of threshold resolves most of the confusion. But a full understanding of threshold requires consideration of the slow manifold. And as I said, append SI as a second slow variable, study the separatrix in the beta SI plane. Okay, so I think that's all that I had to say. Thank you for your attention. I'll be happy to take questions.